Society of Aerospace Medicine members to the position of President-elect of ISOM, which he is going to take up in the summer of 2023. Dr. Usmani's research themes are inhaled drug delivery, airways physiology, lung imaging, respiratory pharmacology, and telemonitoring. And our second speaker will be Dr. Heino Stass. Dr. Stass is a former senior clinical pharmacology expert at Bayer AG. And currently, he is a senior clinical pharmacology consultant in the area of clinical PK/PD for Bayer. And he assumed this position after his retirement earlier this year. As a senior clinical pharmacology expert, Dr. Stass was responsible project leader for various anti-infective development products, including development of inhaled therapy. Dr. Stass has over 36 years experience at Bayer. And our third speaker is my colleague, Dr. Ross Walenga. Dr. Walenga is currently a chemical engineer at the Division of Quantitative Methods and Modeling at the Office of Research and Standards in OGD at FDA. Dr. Walenga received his Bachelor's of Science degree in Aerospace Engineering from Virginia Tech University, and he earned his PhD in Engineering from Virginia Commonwealth University. Dr. Walenga's research and interests include computational fluid dynamics modeling of orally inhaled, nasal, ophthalmic, and dermal drug products to answer questions pertaining to bioequivalence. And our fourth and last speaker of the afternoon is Dr. William Ganley. Dr. Ganley is the head of computational pharmaceutics at Nanopharm. He holds a master's in chemistry from the University of Southampton and a PhD from the University of Bristol. Upon his graduation, Dr. Ganley took up a postdoc position at the University of Bath, where he conducted in vitro characterization of inhaled pharmaceutical products and developed in silico physicochemical, pharmacokinetic, and statistical models to improve the clinical relevance of in vitro tests. Dr. Ganley joined Nanopharm in 2019 to lead the computational pharmaceutics group. His team develops PBPK, CFD, PBPK, and statistical approaches to accelerate and de-risk OINDP development. Dr. Ganley also leads the development of Nanopharm's in vitro in silico approaches, combining realistic testing with simulations. And without further ado, I would like to welcome our first speaker, Dr. Usmani, for the first talk. Thank you so much.
Here we go. I hope you can all see that. And what I'm going to do today really is present a case study on the total and regional lung delivery of salbutamol in subjects with idiopathic pulmonary fibrosis, and really focus in session 2 on the in vivo aspects, and really as a clinical academic, pull out some of the aspects related to lung deposition, which is work that my group's been involved with, as well as on clinical endpoints and pharmacokinetic data. So this is my faculty disclosure. And so if we start with idiopathic pulmonary fibrosis, and I think it's a really good disease to consider. Traditionally, we always talk about asthma and COPD, but why? Because the problem with idiopathic pulmonary fibrosis is in the distal small airways. So there's a need for regional targeting. On the chest X-ray on the right-hand side, you can see a haziness on the right and the left in the lower zones of the lungs, and that's suggesting that there's a problem in the parenchyma, and this is actually the distal lung region where you have myofibroblast proliferation. So it's a good case to think about the need to target the drug into this region. So the current therapeutic approaches, well, they're oral tablets, and we recognize with many oral tablets, for example, oral corticosteroids, oral absorption, hepatic clearance, but they lead to side effects. So nearly 20% of patients have to discontinue the treatment because of adverse side effects, and via the oral route, there is a very poor lung dose. So the proposed therapeutic approach in idiopathic pulmonary fibrosis, and there's a lot of drug discovery and development here, has been by the inhaled route. So we all recognize in our community of the increased deposition in the lung and the decreased dose to the systemic circulation, and thereby improving the therapeutic index. And here are just two examples. The top one you see here, repurposing an inhaled corticosteroid and LABA combination for idiopathic pulmonary fibrosis. And on the bottom here, actually a drug discovery on an inhaled small molecule, an integrin inhibitor targeted at the pathology of idiopathic pulmonary fibrosis. So there's a lot of interest in delivering via the inhaled route in this condition. Now, we've touched on this earlier on in Brian's talk, but I put this here. Well, what's the fate of an inhaled aerosol from the mouth all the way, as he said, downstream into the pharmacokinetic blood region, and the factors that can modulate the deposition and the absorption? And we recognize the aerosol deposition can be affected by the electrostatic charge, particle size, inhalation flow, the airway morphology, disease, the airway disease, and we'll come back to that a little bit later. And then drug retention can be affected by the properties of the drug. We've heard about mucociliary clearance, the thickness of the epithelium, the mucous barrier, surface area, bronchial pulmonary blood flow, metabolism, lipophilicity. And this whole process of inhalation, deposition, dissolution, target engagement, distribution, and the systemic circulation, and we're measuring the blood in PK, and I'll come back. So our group has been focused on particle size, and using particle size as a mechanism of targeting different regions in the lung, understanding the response of the lung, and then looking at PK, looking at pharmacokinetic outputs. And a few years back, actually, as part of my PhD, we looked at the role of different drug particle sizes, monodisperse, and I'll talk about that in a bit, in patients with asthma. And we talked about doing these studies in patients with disease. Now, visually, I hope you can appreciate that smaller particles give you better total lung deposition, and you can see more of the radiolabel. So this is gamma camera images of technetium labeled salbutamol, monodispersed salbutamol. You can see in the 1.5 micron that aerosol is distributed throughout the lung, central and distal lung region. In contrast, look at the 6 micron. There's low total lung deposition, TLD, and the penetration index is poor. So it's concentrated in the central regions, but it's not able to target throughout the airway tree. So this comes back when we're thinking of a disease, and asthma and COPD both require targeting to the large and small airways. So, and I'll come back to that a little bit later. So when we talk about aerosol dispersicity, polydispersed aerosols are the aerosols that are commercial, that are used in the clinic, metered-dose inhaler DPI nebulizer. There are varying particle sizes within the aerosol cloud. There's a wide distribution of particles, and you can see an example here centered around an MMAD of 3 microns. Overall, there's less mass in the respirable range. Now, on the right-hand side, monodispersed aerosols, well, they're in the research premise, they're scientific aerosols. They have a GST of less than 1.22, which defines them as monodispersed, and therefore there is a uniform particle size in the aerosol cloud. So there's a narrow, discrete distribution. So you can actually say that you are eliciting a 3 micron response in the airway when it's delivered by a monodispersed form, and the majority of the drug mass is in the target band. So the aim of our study was to investigate the total and regional lung delivery of salbutamol in subjects with idiopathic pulmonary fibrosis. We wanted to assess the effect of particle size, dispersicity, of salbutamol. Now, salbutamol was used as an inert compound. We're not expecting to see a PD response in our patients with IPF, but we were looking to see patterns of deposition, and it can be easily measured through pharmacokinetics. So we looked at lung deposition using gamma scintigraphy and pharmacokinetic profiling in order to understand the viability of the inhaled route as a potential for idiopathic pulmonary fibrosis. So what was the study design? Well, there were 4 aerosol interventions, and I'll talk you through it. All of them, all 4 periods had charcoal block, and we'll talk about that a little later. And then in period 1 and 2, we used monodispersed aerosols delivered by the spinning top aerosol generator, the STAG, 1.5 microns and 6 microns, and this was at 50 micrograms. And we undertook pharmacokinetic data and scintigraphy, pharmacokinetic in the blood and also urine, and I'll come back to urine in a bit. Then period 3, again, charcoal block, here a nebulizer, Parry, 3.1 micron MMAD with a GST of 1.8. And here the dose was 2.5 milligrams in 2.5 mils, which is the standard dose used. And here, and again, we undertook pharmacokinetics and scintigraphy. And then on the right-hand side, a polydispersed meter dose inhaler with spacer with an MMAD of 2.8, again charcoal block. Here this was unlabeled at a 400 microgram dose, and we took blood, PK, and urine. And this was in 9 subjects overall with idiopathic pulmonary fibrosis. So if we reflect on charcoal block, we will appreciate this. Charcoal block to stop oral absorption, and therefore the PK is reflective of the lung dose. So we undertook salbutamol blood sampling at the time shown, and we also did urine sampling, where 0 to 30 minutes has actually been described to accurately reflect the lung absorption and being independent of the swallowed dose. And that was by Henry Christian's group almost 20 odd years ago, if not more. But here we did do gamma scintigraphy, so we do have an idea of lung absorption. So 2 caveats here. We recognize in some instances no need for charcoal block, as the oropharyngeal route is negligible towards the oral bioavailability with compounds such as fluticasone propionate. And the key point here is in our patients, they ingested variable volumes of activated charcoal. Some could only tolerate 10 mils, whereas our target was 240 mils, and some were able to do that. So we need to bear that in mind when we are talking about charcoal block and whether it was effective, totally effective. Quite a few were only able to ingest 10 to 40 mils. So this is a spinning top aerosol generator. This is how we generated our monosperse aerosols. So in essence, you have a spinning disc driven by compressed air. Salbutamol sulfate in water and ethanol is fed to this disc. A film of liquid spreads around. And with this spinning top, you generate 2 particle sizes, a primary particle and a secondary particle. And the secondary particles are removed by an exhaust. The red box shows you we undertook solid validation in the MMAD through Anderson cascade and also an aerosol particle sizer, that the aerosol was truly monodispersed, that it was stable over time and didn't change, that the drug concentration was reflected at the delivery at the mouth, and most importantly, the radio label did not alter the aerosol profile. And I think this is really important when you're doing radio labeling studies. And we showed that using the Anderson cascade impactor. And on the right-hand side, you can see examples of the scanning electron microscope images of our different particle sizes. The other point I want to make is, what do we talk about when we mean central versus peripheral, particularly in gamma scintigraphy? And I know this is going to be touched on in session 2 today and tomorrow. Well, we in our group looked at different methods. In the bottom, you can see in this 6 different methods to try and work out how how they were different and the variability. And really, the go-to document is the one that's shown there on the top from the International Society of Aerosols Medicine. It's a position statement, a standardization document on how gamma scintigraphy should be undertaken. On the right-hand side, how inner and outer should be assessed. And really, this will then allow us to compare across different studies in different laboratories. So let's now look at our results here. So the images show you the 1.5 and 6 monodisperse, and on the right-hand side, the nebulizer. And if we look at the table for a start, this is a total lung deposition as a percentage of the administered dose. And you can see for the 1.5, the mean was 65%, for the 6 micron, 50%, and for the nebulizer, only 8%. So when we look at the bar chart, that's depicted there in the first column there in blue. We recognize that with the 6 micron, there was more throat deposition, and that's the red hatched bars. And that's probably because of inertia, impaction, and greater momentum for the larger particles. Importantly, there was a lot, if you look on the blue bar right at the end on there on the right, the mouthpiece, the nebulizer mouthpiece content was significant. And that's the reason why there was very little drug being delivered as a dose to the lung. And the other point I want to point out is in green, in vitro modeling has shown in 20, 30 years ago that you exhale a lot of small particles, but actually in vivo, you don't see that. And you can see that the 1.5 micron is only about 10% that was exhaled, as opposed to the values that we see in the literature of 60 or 70%. And we see that across different diseases. So if we now look at the penetration index, so let's look at the table on the right-hand side. This is the inner I and outer O regions as a percentage of the administered dose for each subject. And you can see for the 1.5, you've got good inner, and you've also got very good outer, 40% there. If we look at the 6 micron, good inner, but maybe not so good outer. And with the nebulizer, you can see there's equal balance between the inner and outer, but it's reduced as a percentage of the administered dose. So if we look now on the graph on the left-hand side and we look at penetration index corrective of lung ventilation, well, the 1.5 has greater penetration, as you would expect, to deposit throughout the whole airway tree. The 6 micron lower, as you would expect, because of the larger particle size, impaction, momentum, inertia, and the nebulizer was in between the two, probably more towards the smaller particle. So if we now look at the PK, plasma salbutamol concentration per treatment group, and that's shown in different colors, we can see and actually, probably as a function of dose here, the nebulizer, you can see greater Cmax there with 2.5 milligram in green, followed by the blue, the meter dose inhaler, 400 microgram, and then followed by the monodisperse aerosols of 1.5 and 6 microns at 50 micrograms, which were following each other. But when we dose normalized the plasma concentration profiles to a nominal dose of 1 microgram divided by the amount of drug administered each period, you can see that that flipped. So there you have then the Cmax higher for STAG and 1.5 and 6, the monodisperse aerosols, whereas the polydisperse aerosols, the nebulizer and the meter dose inhaler, the dose normalized plasma salbutamol concentrations were lower. We also went on to compare the MDI plus spacer in our patient group of idiopathic pulmonary fibrosis to those seen in healthy volunteers and asthmatics. And those are the references given in terms of the predicted PK profiling that we looked at. And so you can see that the Cmax was probably similar between the healthy subjects and asthmatics, but was somewhat lower in the IPF, idiopathic pulmonary fibrosis, shown in blue there. And that the elimination phase in IPF and healthy subjects was similar, but this was faster in the asthmatic. So again, bringing out differences in disease, differences in biology that may affect our downstream PK data. Here we see the table of the plasma and the urine. I'll show urine pictorially in the next slide, but plasma, we can see if we look at the Cmax in the red box, is as we've seen, with being higher for the nebulizer, probably as a function of the dose, then the MDI, and then the polydispersed aerosols. You see that right on the far right-hand side there in the red box. And in terms of the percentage of total dose, the amount of salbutamol in the urine after 30 minutes was similar between the monodispersed drugs of 1.5 and 6 microns and higher than the nebulized delivery, indicating higher lung exhaustion from the monodispersed STAG aerosols. So in the final sort of 4 or 5 minutes, then really, what really happens to the fate of the drug? And I think we can divide it into these 3 areas: the physicochemical biological properties of the drug, the regional airway deposition of the different particle size aerosols, and the regional airway factors modulating drug absorption, leading to, and you see that in the yellow arrow there, pharmacokinetics, the downstream endpoint that we're using. And I think one thing, and Brian talked about knowledge gap, and I know Robert mentioned new threat models, but there is, just reviewing the literature, I think we really do not understand the dynamic change in the oropharynx that happens during inhalation that affects the downstream plasma and plus or minus urine PK. And I think it's huge, and it varies between gender, it varies between culture, and there's certainly, this is work that needs to be done. The dynamic change during an inhalation maneuver, particularly across different resistance DPIs from low going up to high. And the other point I wanted to touch on was clinical endpoints. We've heard about this earlier on, and I would say be careful about baby and bathwater. So here we showed that in our original work that even though we had more deposition with the 1.5, the FEV1 was greater for the 6 micron. And the reason is that's because FEV1 is targeted to assess a response in the central region. It's very poor at picking up changes throughout the airway tree. And really, it's a very crude marker. So it's good for the first 5 generations, but it really doesn't tell you much beyond generation 5, going down to 16, and then beyond 16 to 23, which is the alveolar region. And we're talking about regional deposition here, aren't we? So it's not great. So we've undertaken work to understand how we can probe the deep lung, the different regions, the proximal, the intermediate, and the small airways. Time doesn't permit me to go through everything here, but you can group them into imaging, biology, and physiology. And these are 2 recent publications that we've undertaken. And if we just become a little bit more granular, the yellow is showing you the respiratory physiology assessments, the blue is showing you how you can sample the airways, the pink is showing you lung imaging, and you'll hear a little bit about functional respiratory imaging a little bit later. And then there are questionnaires, and the measures there are on the right-hand side that give you an assessment of the small airways. And this is a shout out for oscillometry, which is a non-invasive tidal breathing maneuver that can assess lung mechanics in children as young as 2 and in elderly patients up to 80. And this is where we see the problem with spirometry. And we heard earlier on, didn't we, about FEV1 and spirometry, that this is a forced maneuver and children find it very difficult to do, and so do elderly patients. So I really think that this is going to evolve. Oscillometry is going to evolve, and it can give you an assessment of large, intermediate, and small airways. So in the final 2 or 3 slides, we've done a state-of-the-art review that links the pathology and pathogenesis of small airways to pharmacology, to physiology, to pharmaceutics, optimizing the delivery and the formulation, phenotyping, the path to regulatory acceptance and prospects for the future in asthma and COPD, but really in terms of what's needed going forward. So my final slide before my summary, this is just to tell you that we've got the International Society of Aerosols in Medicine Congress, which is August the 26th to 30th in Saarbrücken in Germany. And I think in summary, we've used particle size to interrogate the airways to understand pharmacokinetics, where we've shown in this particular study that small monodispersed particles achieve significantly better total lung deposition than larger particles, where polydispersed nebulization was poor. That small monodispersed particles achieve significantly better total lung penetration. You saw that with the images as well, compared to larger particles. And that polydispersed nebulization showed lung penetration probably a little bit more similar to the smaller particles, but not as much. And that higher dose normalized plasma salbutamol levels were observed following the monodispersed 1.5 and 6 compared to polydispersed meter-dose inhaler inhalation, while lower plasma levels were observed following the polydispersed nebulization. So we've done work on inhaled monodispersed steroids. I presented that at RDD about 8-9 years ago, but really I hope this opens this session with respect to the further talks, teasing out aspects related to this session. So on that note, I'll hand you back, and I'll hand you back, I think, to Ross. Thanks. Thank you very much.
You. Yeah, first of all, thanks for the nice introduction, Rukia. I have no disclosures to make beyond what you said. I'm coming from industry, and I'm glad to be here after a long time without direct interaction with other experts, and therefore I'm glad to be able to present you the development project that we had for the antibiotic ciprofloxacin. I worked in clinical pharmacology, and we understand ourselves in this development area as the link between the preclinical and the clinical development, and we are trying to generate data which help the clinical developers to or which guide the clinical development in their program. And those data, with a special focus on scintigraphy and in combination with charcoal block, I'd like to present to you now. The next one. Okay, the indication in which this project was developed was the intermittent therapy to reduce the frequency of acute exacerbations of non-cystic fibrosis bronchiectasis. The final dose that we came up for the phase 3 was then testing 32.5 milligram ciprofloxacin as a dose of 50 milligram DPI powder on a BID of 14 or 28 days on-off treatment in this patient group. In the preparation of this program, we had also data generated in cystic fibrosis patients, healthy volunteers, and in COPD patients, and they got us quite some important information to help guide the clinical development. Treatment concept was to develop a preemptive treatment on top of the standard procedures valid in these patients, with the intention to prolong the period between two exacerbations in these patients. Usually 2 or more exacerbations per year exist, and each time the patient experiences this, the lung function deteriorates until either they have to die or get a lung transplant. So the relevant pathogens, they have minimum inhibitor concentrations of 4 micrograms per milliliter, and ciprofloxacin is an ideal candidate to treat those, but it was not developed for inhalation. As you all know, it's a long time used as IV and oral. So the PKVD concept we tried to realize with this drug was to achieve kind of a depot in the lung, and proving this is really a real big challenge. We wanted to maximize the drug concentration, epithelial lining fluid, but also in mucus plugs, and with the intention to compensate losses. Again, here it's local targeting, so that we had to try to reach homogeneous distribution in the lung to get the alveolar space covered, plus the entrance door for bugs closed, so that frequent infections of the lung can be prevented. So yeah, the PKD parameter relevant is were AUC of MSA in the target, not in the bloodstream, and time above MRC, keep it as high as possible for as long as possible to really shut this off, and at the same time minimize systemic burden to avoid systemic side effects. Yeah, the drug device combination that we chose for this is shown here. The drug itself, as I said, has a BCS classification depending on the literature you're looking at, of 2 or 4, quite a decent bioavailability. However, transport characteristics of the drug and solubility characteristics of the drug are important. At physiological pH, the solubility ranges at a level of about 200 to 300 milligrams per liter. The challenge that we faced here, we wanted to reach much higher concentrations in the lung, and we did some investigations in samples. Again, the lower graph on the left-hand side of the graph, which showed that we have quite a solubility limitation there, which is different. It ended up at a level of 120 micrograms per milliliter. And we have a kind of a different unbound fraction, which is nonlinear when it comes to concentration, total concentrations of the drug in these matrices of above 200 and more. Again, this heavily nonlinear behavior is a challenge in terms of PKPD guidance for such program. The formulation, the powder formulation itself, was based on a platform developed by Novartis using highly porous spherical particles coated with modified phosphatidylcholine molecules, which were designed to prevent aggregation and to guarantee de-aggregation when it comes to the inhalation process. Mass median aerodynamic diameter was in the range where we expected quite good distribution in the lung area. Fine particle fraction of 51% around about should give us a good range of lung deposition there. The inhaler itself is a handheld breath actuated inhaler, again based on technology by Novartis, required based on preclinical in vitro experiments, 45 liters per minute peak inspiratory flow and 1.3 liters of inspiratory volume. Sorry, yeah, these experiments were then confirmed in the clinical study in COPD patients, in non-CF bronchiectasis patients with all decrease of lung function impairment that we could get hold of down to 25% of. In terms of peak inspiratory flow, the criteria were met with one inhalation process. In terms of inspiratory volume, to be on the safer side, it was advisable to get us 2 inhalations per dose. And this was also the recommendation taken forward into phase 3. Now, the further program for the biopharmaceutical support of our PKPD concept consisted of a variety of points. Quantitative deposition patterns were set because we wanted to know that for various calculations across different study populations and across different lung function ranges. This was also in the scintigraphy charcoal plot study, the primary parameter to test. But equally important was for us the bridging from healthy subjects into the patient population, which was consisting of regional deposition, influence of the pathophysiological factors on lung exposure, difficult to assess, and therefore we kind of used a method first looking at expectoration in order to come to an idea of what are PKPD relevant lung concentrations which we could use for further PKPD calculations. In this context, we were interested also in the role of the charcoal block method as complementary method to the standard armamentarium that you find in the literature, and especially the value of PK sampling from the lung, because we were definitely interested in getting lung samples and concentrations. So scintigraphy was set, but the question was lung PK data. What we did is in various patient populations look at drug loss due to expectoration. When you look at this graph, this summarizes study results from around about, I don't know, 12 or more phase 1 studies in patient populations. Cystic fibrosis patients obviously can bring up by physiotherapeutic methods up to 20% of the drug when it comes to expectoration. And in COPD, there was, though absolutely not significant, but one could assume that, first of all, the inability to clear the drug by epithelial clearance, the mucociliary clearance, is compensated subsequently by the expectoration. It's, as I said, statistically not significant because this is an absolutely high variability that we saw there in these studies. Second point, which is, in my view, a very important point, not only for the drug development, but could be an important factor for bioequivalence approaches for these drugs. When we increased the dose in the dose escalation step, because we wanted to see whether we can prolong the duration of the high drug levels by increasing the dose, what we saw is that all of a sudden the expectorated amount increased, and we did a calculation. It's in about the serum plus that we put into the formulation, which the expectoration brought out again. So our take home for this, for the development, was it doesn't make sense to go with higher dose. More doesn't help more. Dose frequency is the solution to that, and BID treatment was then the take home from that. Dust load maybe the reason for that. This could be a point for in BE studies when the formulations differ from each other and patients tolerate less due to the underlying disease. And yeah, changes in formulation in an ongoing program. The second point was, as we were interested in collecting lung samples anyways to see what happened there, we performed PBPK simulations using data from the studies in order to see what is the effect of sampling on the pharmacokinetics from the lung vacuum measure in these patients. And if you see, the earlier you get a sample, the heavier the amount is. And as you can't control the sputum volumes that you get when you induce sputum or when you collect sputum, you see that in this scenario simulation, the effect can become bad. And therefore, in the phase 3, we thought for confirmation of our PKPD concept, stay away from collecting lung samples unless you get spontaneously sputum brought up. And we use this in combination with POP-PK approach to confirm our those decisions. And this is also when you consider BA concepts based on lung sampling, either if you use bronchoalveolar lavage or induction of sputum, I guess self-speaking that at least for ciprofloxacin, it's not a good idea to do. Now to the combined scintigraphy and lung and charcoal block study. This is the scheme that we applied in the charcoal block study. 20 grams of activated charcoal immediately before the inhalation, and then 2 hours later and 4 hours later again 10 grams of activated charcoal to really be on the safe side and get the absorption, the oral absorption really stopped at this. This method, in our view, had a lot of advantages over the rest because other than the scintigraphic model with kind of a snapshot showing what happens immediately after drug inhalation, but not being able to assess the lung kinetics, this this method would indirectly give us data over the whole dosing interval and taking into regard what happens with a patient when expectoration takes place. Nevertheless, for the biopharmaceutical development, we use this method only in the healthy subject population. But in principle, this is a big advantage, and it avoids technical bias when you avoid a PK sampling in the lung. Yeah, and in combination with the scintigraphic data, we have also tried to perform extended pharmacokinetic calculations. Fortunately, in this case, the nonlinear behavior and the other kinetic data made the guidance of the development program relatively easy, and because we could cut it down to a relatively simple in vitro kill kinetic experiments in order to get information to the clinical development on the way forward for the clinical development. For BA studies, this would not help. There, the effort is a bit bigger. It would need their PKPD modeling approaches, informing the model with the data that you get and the charcoal block data and the scintigraphic data. One word of caution: the ciprofloxacin drug has is a transporter substrate, and therefore the charcoal block might be biased because it interrupts enterohepatic cycling of the drug, or and therefore the amounts calculated by charcoal block coming from the lung might be underestimated, depending on what. For moxifloxacin, this has been shown in a study investigating the effect of a charcoal block for IV, and there the bioavailability was decreased by 20%. So that's an important point to consider. For ciprofloxacin, we estimate that this effect might be minor, especially because this was not a pivotal study designed for confirmatory. It was an experimental one. Therefore, we could live with the underestimation that we have here. But for BE studies, again, recommendation would be consider as part of the design of the study in IV arm, and ideally even IV arm plus charcoal, because with this you can quantify the amount taken away from the systemic circulation by enterohepatic cycling. The study design, by the way, do I still have time?
You do 3 more minutes.
Okay. Okay. Yeah, this was the design. So the healthy subjects were in a crossover design in this study, and NCFB patients and COPD patients were a single dose treatment before inhalation. We did a krypton contour scan, and during the inhalation, then the scintigraphic assessment using technetium as the label. These are 3 examples that we got from the study. On the left-hand side, healthy volunteers. Data show nicely the lung is not only covered centrally, but also in the peripheral part. So the distribution is quite decent there. You could clearly discern between lung exposure and oral absorption. The stomach was clearly seen. Patients with severe damage in the middle, the NCP patients, where a significant part of the lung was really damaged, also had coverage of all ventilated areas. And a COPD patient, unfortunately can't see because had a very poor lung function. Also there, the coverage of the lung was good. And we used then these data to quantify deposition patterns. Results are shown here. Surprisingly, relatively good variability. Interpulmonary, around about 50%. Extrathoracic deposition, around 40%. Really nice intra-subject variability could also be derived. It's around 50%, and unfortunately a low percentage of the drug which was exhaled. This is especially important for environmental burden assessment of the drug. Yeah, the nice thing about it that we could conclude, independent from the disease, the quantitative fate of the drug after the inhalation could be determined. Even individually can use these data and inform the PPPK model with these data individually. The regional distribution was also important to know for us, and it could be easily calculated from these data. The concentration time profiles in plasma that we got out of the study are shown here. We saw a 41% exposure reduction by the charcoal block in this study, and the profile, the black one here, represents the charcoal block profile. Resembles a little bit more a profile of an IV, because with the first dose we kind of reached the peak concentrations, whereas the red profile without charcoal block really is biased by the oral absorption, which is quite considerable, as you have seen in the data. So the charcoal block profile was then used to really do further PK/PD calculations. And it's what you can do with this. We did this also for other projects. You can deconvolute lung absorption profiles. From this lung absorption profiles, you can derive amount over time profiles in the lung, and based on physiological parameters, even derive concentration time profiles in the lung for healthy subjects or patients. And this would be at least a surrogate which is clinically providing clinically relevant PK/PD information, which could use to determine doses. And with some more efforts, maybe even for BA considerations. A little bit calculation with and comparison of charcoal block and scintigraphic data are shown here. Left-hand side again, the data which I already showed you, scintigraphic. What you see is that by the charcoal block method, the nominal dose taken up into the systemic circulation, it's 30% of the nominal dose that you administer is taken up into the systemic circulation. This apparent difference, however, can be explained that the charcoal block method would cover for the mucociliary clearance effect that you have in healthy subjects, and by it's tempting to speculate that the 20% that we saw in the cystic fibrosis, which is kind of a mucociliary clearance block situation in these patients, is representing what we see here in the charcoal block. So really decent data which get us a deeper understanding of lung kinetics compared to the classical approaches. And I think then I'm already at the conclusion part. My personal opinion is that this combination is really a useful instrument, at least in case of the CIPRO DPI, where the technical limitations were minor, to get the data that you need for such investigation to guide clinical development. But the real advantage is that the combination with PBPK modeling would bring us forward and get us much more information on real PK concentration time profiles in the lung. I think the rest, I don't need to repeat that. But this, thank you very much for your attention.
Hello. Today I'm going to be speaking to you about model purpose and selection for supporting development and approval of generic locally acting orally inhaled drug products in the United States. So considering bioequivalence or BE at the site of action, for locally acting orally inhaled drug products or OIDPs, lung tissue is the site of action. So there's this figure on the right, and this has been talked about a couple of times today, so I won't belabor it, but essentially when you inhale your drug product, it's either going to go to the gut or it's going to go to the lung. If it goes to the gut, it's subject to the first pass effect. If it goes to the lung, then the free drug will dissolve, permeate into the lung tissue and then into systemic circulation. So, considering BE, the challenge here is that you have regional deposition on one hand, which is something you can measure with a variety of techniques, and systemic PK, which you also can measure. But really, one is upstream, regional deposition is upstream, and systemic PK is downstream. So it creates a bit of a problem for how do you determine BE. And this has been mentioned a couple of times before, so again, won't belabor it, but FDA has developed a weight of evidence approach to handle this problem using a variety of tests that include a comparative clinical endpoint or a pharmacodynamic study. And as mentioned this morning, beautifully by my colleague, Yuqing, there's certainly problems with variability of these studies, and also, like Dr. Ward mentioned, there's problems with expense of these studies. And so what do we do about this? So we've been considering various alternative BE approaches for OIDPs, and thinking about BE at the site of action. There's several studies that have been mentioned. You've got your realistic mouth throat, or MT, APSD testing, which can tell you something about drug that's being delivered to the lung and give you some assurance of regional lung deposition. Dissolution, which of course can tell you something about how it dissolves, hopefully biorelevant in vivo. Morphology, which also can tell you information about what's going to happen to the drug after it deposits in the lung. Spray velocity and evaporation, which tell you something about the aerosol prior to deposition in the mouth, throat, and the lung. And then you come to PBPK modeling, which can consider many things all at once, can tell you about the interplay between deposition, solution, permeation, absorption to lung, absorption to systemic PK. So it can really help answer some questions related to that interplay. And then you have computational fluid dynamics, or CFD, also semi-empirical regional deposition model, just another kind of flavor of the same thing where you're predicting regional deposition. So the question then is, how can the available alternative in vivo, in vitro, and silico studies be used to ensure BE at the site of action? So considering all of these, it's my opinion that assurance of total lung deposition, regional deposition, and dissolution are all needed. So just focusing on regional deposition here, how is regional deposition best characterized? So we saw a couple of talks here from Dr. Usmani and Dr. Stass about using in vivo techniques. So gamma scintigraphy is one potential technique, uses a 2D image from a radio-labeled aerosol and can give you an estimate of central to peripheral ratio for lung deposition. And I've got this figure here that Dr. Usmani also showed this morning. And it does highlight one challenge, is that different groups could define the central and peripheral regions differently. So you can have some variability from study to study. Now, if you do something like Dr. Usmani suggested, if there's a standard that could help with that. But as of now, you have different groups that may do different things. There's also single photon emission computed tomography, or SPECT, which can be combined with computed tomography, or CT. This provides three-dimensional information. Tomorrow, Qinglong Lin will be speaking about regional deposition validation, and his group has done some SPECT-CT studies. So regional deposition can also be measured in vitro. And one way is just using a realistic mouth-throat model, because you use this rapid prototype model with HPLC to measure the drug. But you can also include tracheobronchial branches, and that's been done before using a few of the larger airways, but it only goes typically only so far, only a few branches. However, there are some newer techniques that have been introduced in literature that use an entire whole lung. So obviously, if we could measure whole lung deposition in vitro, that would be a wonderful technique. But it is a new concept, and as of yet, I would say it's not necessarily mature, but something to kind of keep an eye on, I think. And then coming to in silico methods, how can you measure or predict regional deposition using in silico methods? So one technique is CFD. This allows for prediction of fluid and particle transport. And this is just a figure on the right showing how something like this looks like, the results might look like, where you see the particles deposited in the various lung models. And one real big advantage is that it allows for consideration of realistic geometry. So you can have actual patient-specific geometries. You can consider the actual device in your model. And it can consider the effect of input, plume geometry, and APSD. Now, there's also semi-empirical models which use an algebraic formulation. And these, the way these work is they predict branch-specific deposition using formulations for impaction, sedimentation, diffusion. You sum these together and you get branch-level deposition. And these can consider the effect of input PSD. So they're not set to consider plume geometry, but as with CFD, they both can consider particle size distribution. So then the question is, if we want to establish BE with respect to regional deposition, how can we do that? So in vivo imaging studies are certainly interesting, and I think Dr. Usmani had some really interesting ideas about this. But there are some limitations with including a radio label that might alter the drug. But I think he had some good points about if you take the particle size measurements and show that they're not that different, then that you can certainly lessen the impact of that. From what I've seen, it seems to be typically limited to a small study size, and I think that has something to do with just the complexity of the technique. So that could be a limitation there. In vitro whole lung deposition studies, something I mentioned, there's some recent studies that consider this type of technique, but not going into detail for this talk, but I think that they're interesting, but maybe not yet mature. And the one I'm focusing on for this talk, not to say that the other two methods aren't viable, but focusing on in silico regional deposition models that are capable of accurately predicting regional deposition and considering intersubject variability. So our opinion is that there are two potential regulatory pathways using modeling. Doesn't mean that these are the only two, but these are just two that we've identified that we think are viable. So the first pathway would be if you're taking your regional deposition model and then using it to establish BE. So as part of your kind of a direct evidence as part of your BE approach. So in this case, your realistic mouth throat APSD testing is used to establish bioequivalence according to total lung deposition and can provide some reassurance of regional deposition because you are measuring particle size as it enters the lung. Then the modeling can be used to establish BE according to the regional deposition predictions. The solution we think is an important part that helps you understand how that deposited particle behaves after it deposits. And then the other studies that I mentioned, morphology, spray velocity, and evaporation rate studies, they provide some additional assurance of sameness, and they touch on some of those other aspects. So what kind of model could I use to support this particular pathway I'm speaking about now? So I mentioned CFD. That's a high fidelity modeling approach. It might be more likely to reflect in vivo imaging data just because it can consider the plume geometry and the complicated physics in the mouth throat. But disadvantage is that it is resource intensive and it does require specialized training. It's not something you can just jump into. You have to take a significant amount of time to learn it, or find an expert who can help you. Just a side note is that you can combine it with another technique called discrete element method or DEM modeling. And this can be useful for understanding particle interactions in dry powder inhalers like agglomeration processes and deagglomeration processes. And another thing to mention is that it can consider intersubject variability. So if you have a variety of lung models, that would be the way to do this. I also mentioned semi-empirical models also can be useful for this pathway. This is a simplified model that gives you potentially still adequate comparisons with in vivo imaging data. It is a faster modeling approach. It's easier to execute, requires a lot less training. So that's certainly a big advantage of this approach. And also can consider intersubject variability if you've got the sufficient data to tell you the lung geometries of different patients. And also touching on PK modeling. So PBPK can be useful in this scenario, this pathway, for understanding the relationship between regional deposition and absorption. So especially if you've got a drug like fluticasone propionate that's very slowly dissolving, regional deposition is not necessarily going to tell you all you need to know about regional absorption. And so coming back to this idea of using deposition prediction to support your BE pathway, one particular way of doing this, calling it a population modeling approach. And so Dr. Usmani's group actually did a study a couple of years ago using functional respiratory imaging technique to predict regional deposition, and they did this in 20 patient-specific geometries using CFD. And one thing that this technique allows for is that not only do you have the patient-specific models, but it can identify the patient-specific inhalation profile. So it gives you like a greater sense of the intersubject variability. And the way they completed the study was that they compared the regional deposition predictions from 2 different MDIs, much like you would do for a bioequivalence study. So another way of doing this would be to use a representative model approach. Tomorrow we hear a talk from Dr. Qinglong Lin about their modeling approach. This was a funded project from FDA to University of Iowa. And what they did for this study was they had a CFD model, and they had 2 healthy subjects and 8 asthmatic subjects. Healthy female, which you see on the top right, HF. HM is healthy male. Cluster 1, cluster 2, cluster 3, cluster 4. And each of these clusters, he'll talk more about it, I'm sure, tomorrow. These clusters were identified to be representative of different kind of subpopulations of asthmatic patients. And if you look at the results, which were divided among 4 different particle sizes, 1, 2, 4, and 8 micrometers, and then the lobar deposition was predicted, you can tell that there's quite a bit of variability from one representative subject model to another. But if you take this approach, you've got your representative models. You don't necessarily need any more if you can be convincing that these are representative of the population that you're the population you're after. Okay, and for semi-empirical regional deposition modeling, there's a paper a couple of years ago from Bo Olsen and his colleagues, and they predicted a peripheral to central regional deposition ratio for 9 different dry powder inhalers, and they compared the results with in vivo gamma scintigraphy from 18 different study arms. And they use a representative mouth throat model, but then they also had a asthmatic model that they constructed using like a consistent diameter constriction. And so this was, I took some of the results from the study and I plotted it here using a line of identity. And it can show you that in some cases, the predictions were very close, like right along the line of identity. Some cases over-predicted a bit. So, but I think that if you're using this type of modeling, and CFD too, really, like both types, you'll just want to make sure that you have a good correlation between in vivo and in silico predictions. So I've talked a lot about regional deposition modeling, but PBPK modeling, I'll touch on a little bit here. There'll be a couple of talks tomorrow from Kevin Fang and Jurgen Belita talking about using population PK and PBPK to connect PK metrics to regional depositions. So they'll have a lot more details on that. But I did want to mention that while regional deposition is important, it might not always be a surrogate for regional absorption. And so PBPK, of course, can be useful for understanding how dissolution and permeation affect that relationship between regional deposition and regional absorption. This is just an example of a PBPK model where you've got the different regions, the bronchial region and the alveolar region, and you have different compartments depending on each region that account for some of the physiological differences. So, okay, so you've selected a model that you think is useful for your approach. One thing I think to consider is how you're going to connect this with in vitro data. So, oftentimes you may have in vitro data from other studies that can help support your study. So if you've got APSD results from mouth-throat testing, these can be used for inputs to some semi-empirical and CFD models, and also can be used to help validate CFD model predictions. Dissolution data also can be used as inputs. And other relevant studies include plume geometry and spray velocity. So spray velocity in particular can be used as both validation and model inputs. And I think the one thing you just want to ensure is that your modeling assumptions are not in conflict with your collected in vitro data. That there's nothing that kind of violates using the in vitro data as an input for your model. Just a brief word on statistical approaches that might be used for this scenario. If you're using what I was calling a representative model approach, if you're just using several representative models, population bioequivalence might be useful because you might have APSD results from, say, 3 lots, 10 units, which kind of falls more in line with how PBE is analyzed. If you're using what I call the population modeling approach, then you've got predictions from several different individuals, then average bioequivalence might be appropriate for that scenario. So I talked a lot about this first pathway. The second pathway that I think could be useful would be to identify a biopredictive study. So again, as before, we think realistic mouth-throat testing could be useful for establishing BE according to total lung deposition and providing reassurance of similar regional deposition. But in this case, modeling would be used to identify biopredictive studies to establish correlations between results of in vitro or in vivo studies with regional deposition predictions. So really, in this case, the in vitro or the in vivo studies are serving as your bioequivalence, your check on bioequivalence rather than the model itself. The model is just kind of supporting your choice of study. And also the model can be used to understand the sensitivity of those results with respect to regional deposition. Dissolution can be used as before to ensure that deposited particles behave the same. And morphology, spray velocity, and evaporation rate studies provide some extra assurance. So selecting models for this pathway, so again, you can use regional deposition modeling, either CFD or semi-empirical models to establish these correlations. If you're trying to draw a correlation with plume geometry, you would probably choose CFD because semi-empirical model really doesn't consider that aspect. And then for PK modeling, like I said, tomorrow we'll have more talks on this, but population PK or PBPK could be useful for establishing correlations between PK metrics and regional absorption. So not just showing that they're correlated, but showing like the precision of how sensitive it really is. Like if you have a 10% difference in Cmax, what does that mean for a difference in regional deposition, quantitatively speaking? So you've chosen a model and you've chosen your model purpose, but one thing you have to be sure of is to establish model credibility. I just want to have a brief word on this. This is a standard that we find to be useful from American Society of Mechanical Engineers. It really developed for medical devices, but I think it's applicable here too. So it's a risk-based approach where you identify the context of use, which is like the question you're addressing and how the model contributes to that. You identify the risk by how much you're using the model as compared with what's the consequence of a bad decision. And then you establish credibility of verification and validation. So one example here is for a CFD model where they've predicted central and peripheral deposition. So in this scenario, you would predict deposition in all the lung regions. And then you would make sure that you define the barrier between central and peripheral regions using some kind of solid justification from literature. And if you've got a model that only extends so far into the lung, you might need a complementary model so that you're predicting through the entire lung. And like I mentioned, I think previously that the preferred source of comparator data would be in vivo imaging data. And in this case, you can see that they had a pretty good comparison between central and peripheral predictions and the in vivo data. So one limitation is, like I mentioned briefly, that generally speaking, in vivo imaging data is from a 2D image. So it will be divided into central, intermediate, and peripheral regions. But there's a lack of precision when comparing, three-dimensional predictions with a 2D space. So this paper that I'm focusing on here, what they did was they had a CT scan and they compared it to these different regions. And they identified, like on a branch level, how much of that branch would be in this 2D C region or 2D P region. So it kind of allows for a way for translating between your predictions and your in vivo data. So, to support that, a small in vivo study with a few subjects could be useful for supporting validation. So you've got both the gamma scintigraphy and the CT scan data to compare against. So a few conclusions. Alternative B approaches in lieu of comparative clinical endpoint for PD studies might include modeling studies. Regional deposition modeling provides a means of establishing BE according to regional deposition. There are two pathways that I described. One, we're using a deposition model to kind of directly support bioequivalence, and the other pathway is using any in silico model to identify biopredictive studies, and then use that to justify your in vitro or in vivo studies to infer on regional deposition. And model selection should consider a variety of factors. You've got the ability of the model, how efficient the model is, how accurate. Of course, model credibility is important, and I talked a little bit about what ASME BMV40 is. And validation of regional deposition predictions is expected to be accomplished using in vivo comparator data. And I just want to acknowledge all the colleagues who have contributed to this work.
Hey everyone, I'm Ganley. I'm head of computational pharmaceutics at Nanopharm. Thanks for allowing me the opportunity to come here and speak today, and also thanks for the kind introduction. I'm also going to be talking about the purpose and selection of models for alternative bioequivalence approaches. And I guess where my talk may differ from Ross's is this is kind of the Nanopharm approach to listening to the science, looking at what everyone's been doing, and putting together kind of a package that we think is, a way of thinking about these things and the sorts of things we've been working on. So over the next 20 minutes or so, first I'll talk about kind of what the goals of the in silico approach should be, then talk about what the toolkit is. I think there'll be a lot of overlap with previous talks in this session on that. Then speak about how in vitro, in vivo, and in silico evidence can work together within an alternative bioequivalence approach. And then I've got two case studies to talk about kind of various important aspects of these approaches. The first one is looking at fluticasone propionate DPI and how you can use a sensitivity analysis within a PBPK model to understand what the important product attributes are and link those to clinical data. And the second one is looking at disease state differences. So this is how you can use modeling and simulation to bridge between clinical data in healthy volunteers and patients to hopefully avoid having to do PD studies in patients altogether. And then at the end, I'll bring all of this together and kind of talk about what we think the future might look like for in silico approaches within bioequivalence for already inhaled drug products. So first slide I wanted to put up in this was kind of just highlighting the definition of bioequivalence. So what we're looking for is to demonstrate equivalence in the rate and extent of exposure of the active ingredient at the local site of action in the lung. So that's something that we need to keep in mind as we're designing these approaches and figuring out what kind of models we need to use. And kind of within that, there are four key attributes that we want to try and figure out, and for each specific indication and drug product, these are going to be slightly different, but if you kind of abstract them to their kind of base level, these are the ones that I think are important. So the first one is the endpoint. What is the important attribute that you're trying to model? Is it deposition in a particular region in the lung, or as Ross said a second ago, is absorption or dissolution in the lung important as well? And is that the kind of key contributor to exposure at the site of action? And those two cases are quite different and may influence what kind of models that you want to use. The next thing is the disease state. So the key thing that we want to do is make sure that the drugs are effective in patients. So are there any physiological, lung geometrical, physico-pathological differences due to the specific disease state we're looking at that might influence your decisions in modeling? And how do you use modeling to represent the disease state in a way that makes it effective and relevant? Then we've got the product. We've talked a lot about how there are now some biologically relevant in vitro tools, realistic mouth throat models, breathing profiles, dissolution studies of respirable doses, morphology considerations for respirable doses as well. What's the variability of these things? How does each dose differ from another dose? When you come to dosing patients in clinic, potentially, how might the product differ from sort of puff to puff? And then, of course, there's the patient consideration. What's the inter and intra patient variability? How might the way that a patient use a device vary? How might a breathing profile vary from dosing events, and how might it vary between patients? And how can you build that into your modeling approach as well to make sure that that is relevant? And in designing a modeling approach, what you want to do is make sure that you bring all of these things together and acknowledge that they will be very different depending on the type of dosage form you're using, but also the type of API you're interested in as well, and of course the disease that you're trying to treat. So in terms of the in silico toolkit, this slide is very similar to the sort of stuff Ross was talking about a second ago. So there are two broad classes of model that we want to try and use for OIDPs. These are models that predict deposition and models that predict pharmacokinetics. And within those, I think there are broadly two classes per category. For deposition, you're talking about semi-empirical algebraic models versus CFD versus or CFD. And in pharmacokinetics, there's bottom-up mechanistic PBPK models where you're modeling individual mass transport processes and kind of building up the story from there, versus population PK, which is kind of more top-down, where you're trying to build a structure around PK data to try and sort of tease out some information about where things land in the lung. But with all of these things, there are some questions that you need to make sure you ask. How do you get the right input data? How do you incorporate patient variability? What external data is required to make sure that your model is representative? And of course, what sort of disease state considerations you need to make if you're looking at bridging between healthy volunteer data and patient and predictions in patients. And as with all of these things, the modeling can't stand on its own. And what we need to make sure that we design into the holistic alternative bioequivalence approach is the way that all of these things come together. And I think Brian put it quite well in his plenary talk earlier, where he was discussing the different sorts of evidence that kind of need to work together. And I think there's an important distinction here between the supportive evidence that might tell you something about how your drug product behaves, but then the kind of the important critical and pivotal evidence that either will serve as a model input or will kind of almost stand on its own as a piece of bioequivalence evidence. So for example, and I like to think of this as kind of a pyramid where you have you build the base level understanding of your formulation and device. And this might come from for example, understanding the suspension characteristics within within the can in the suspension PNDI, or the flowability of bulk powder within DPI. And then you build that up into understanding how the product performs under realistic conditions. So this is where your mouth-throat models, your aerosol capture methodologies come in. You can then use that information to input into models where you're running simulations of healthy volunteers and patients. And I think at the pinnacle of this, and that's something that we haven't necessarily spoken about so far, is the idea of an in silico clinical trial. So what's really important in all of these things is not just knowing your point estimate of kind of what a idealized or typical patient might do and what the average MMAD or the average predicted deposition might be, but it's how these things might vary. Because in the real world, the reason that CCEP and PD studies require such large patient numbers is because the variability is significant. And we've seen lots of examples of that today already. So it's not good enough just to say, well, my MMAD is 2 microns, that predicts a deposition of this. And based on my idealized PBPK model, I'm going to predict a single plasma concentration curve. And from that, potentially infer some local exposure parameter. You need to make sure that all of the different levels of variability and how they interact with each other and propagate through the entire approach come in. And that's where this idea comes. And actually, given the modern sort of techniques and the speed at which you can generate some of this information now, these things aren't as challenging as they used to be, and that's something we definitely need to consider. So in building this entire approach together, you can kind of add layers of understanding, show that you really know how your drug product is working, but then also use things like sensitivity analysis within the models to tease out what the important attributes are and focus your attention in terms of a bioequivalence determination on those and not worry about the things that you can demonstrate aren't so important. So the first case study I'm going to talk about is on fluticasone propionate DPI. So here I'm relying quite heavily on this study that was led by Gunther Hockhaus, who is in the room, and various of the co-authors that are also present here today. And what they did was to manufacture 3 formulations of fluticasone propionate DPI. These were manufactured specifically to have different aerodynamic properties and dissolution rates. These were characterized in a great amount of detail with different techniques, including realistic APSDs and sort of dissolution following aerosol capture and things like this. And then a PK study was conducted. The team was then able to use population PK modeling to connect, or to attempt to connect the deposition in different parts of the lung with the PK data, and then use all of the in vitro characterization that was done to kind of back this up. And it was quite a nice holistic study. So what we did was just to kind of show how a modeling approach could be used. We took some of this data and we used, instead of using kind of a top-down pop PK approach, we used a bottom-up regional deposition PBPK modeling. So the two modeling approaches we selected for deposition and pharmacokinetics here were the algebraic NCRP model. This was a healthy volunteer study, so we were looking at the Weibull lung model. We took breathing profiles from information from the paper to generate idealized breathing profiles, and we did the deposition modeling in the Preludium software. We then took the predictions of deposition throughout the lung from that stage and input them into our own whole body PBPK model. So this is a complete body PBPK model with compartments for each of the different tissue types in the body. All of the tissue types are assumed to be well stirred apart from the lung, which is broken down into all of the generations of the Weibull lung. And then each lung generation is further broken down into lung lining fluid, epithelium and subepithelium. And we have dissolution processes and mucociliary clearance happening in the lung lining fluid. So we think this is a fairly detailed model of the lung that can allow us to make some pretty detailed predictions. So the input data for these were the emitted mass and the APSD. So as Ross mentioned, in the algebraic models, these are the key inputs. For the PBPK model, we took the predicted deposition in each generation of the lung, and that went in. We also had dissolution rates. We had a process of dissolution in the epithelial lining fluid, which covered that process. And actually, to get these rates, we did a model fitting parameter to the PK data from the paper. And I'll show you in a second that correlates quite nicely with the in vitro data. So in terms of parameterization of the model, we put the breathing profiles, particle characteristics, and the lung model into the deposition. For the PBPK model, we were able to take a lot of data from literature and then essentially adjust the model based on some PK data. One of the, I think, advantages of the bottom-up PBPK approach is that because you can predict administration via lots of different routes with a whole body PBPK model, we were able to verify the systemic disposition parameters in the model using literature in vivo data, sorry, intravenous PK data. And then when we came to ensure that the model produced credible predictions via the inhaled route, we were able to fix the parameters that we knew about systemic disposition and only focus on the parameters such as permeability and dissolution in the lung. So that kind of made the model building process a lot more straightforward. So here are the first set of results. So on the right, left-hand side, you can see the points representing the PK data from Gunther and Co.'s paper. The lines are the model predictions. And like I said earlier, we adjusted the dissolution rate for each of the three formulations to obtain a kind of in vivo or in silico dissolution rate. And this allowed us to obtain quite good matches to the clinical data. We then compared those rates to the mean dissolution time and the dissolution rate that was measured in vitro in the paper, and again found really nice kind of sort of in silico / in vitro correlations, which was quite encouraging and showed that what was measured in vitro was actually quite representative. But then the next stage, and I think this is where the real power of the approach came, was we conducted a sensitivity analysis. So this is where we took the model predictions for one of the formulations, and we adjusted a few of the parameters that we thought were quite important up and down. So we doubled and halved each of them. And then we looked at what the effects of these changes to the model parameters would be in terms of the predicted Cmax and the AUC of the model. And bearing in mind that we'd already verified that the model reproduced the systemic PK for these systems, we had a good degree of confidence that these would actually be quite representative. The parameters we looked at were the mouth-throat deposition. So this was effectively doubling or half, well, actually it was only doubling the amount that got into the lung and wasn't swallowed. We looked at doubling and halving the MMAD. So this would affect the regional deposition and also the dissolution rate as well. So on the left-hand side here, you can see that when you reduce the mouth-throat deposition, you see quite a large increase in both AUC and Cmax. This is potentially expected. If you get more into the lung, you're going to see more getting into the blood. What's interesting is that if you look at doubling and halving the MMAD, you only see kind of modest changes. Now, this potentially suggests that the Cmax and the AUC aren't sensitive to regional deposition in the lung. However, I think an interesting additional piece of work to this to do would be to look at what the doubling or halving of the MMAD actually do to the distribution or the deposition in the lung, and then sort of try and see if we can make an assessment following that, which I think will be interesting additional work. And for doubling and halving of the dissolution rate, what we see is that Cmax is quite sensitive to the changes, whereas the AUC isn't. So kind of the total dose getting through isn't necessarily sensitive to the dissolution rate, whereas that peak, that maximum plasma concentration is. Now, of course, FP is a very well-studied system. A lot of this thing kind of chimes with what we'd expect anyway, but this is the sort of approach that we can use for potentially any molecule, any generic product, and to understand what the important factors are and to inform us on what kind of key in vitro studies need to go into the alternative approach, and potentially to refine our in silico models to make sure that they are only considering the most critical factors. So the second case study I want to talk about is something that's a little bit more based around patients and local deposition. So here we're looking at a spray dried formulation of the antibiotic amikacin. This was delivered via the orbital device, which is a high payload DPI. It effectively has a puck inside it filled with powder that orbits and emits the aerosol. We collaborated with Fluida to do some CFD or functional respiratory imaging on a cystic fibrosis lung and a healthy volunteer lung. And what we're interested in here was, first of all, using the CFD PBPK approach to make some predictions of systemic and also local PK, but then also start to see what disease state considerations are important when we're trying to translate information from healthy volunteers to patients, and make sure that when if we were to use this as a piece of evidence to inform regulatory decision making, we're taking into consideration the right facts for the patients we're interested in. So as I just said, the two types of modeling we used were CFD and whole body PBPK. The PBPK model had exactly the same structure as the one that I presented already for FP. We just reparameterized it for amikacin. And the CFD used real measured breathing profiles and, like I said, CT scans of a CF patient and a healthy volunteer coupled to the orbital device. The input data was very much the same as before. The CFD, of course, takes a few additional inputs compared to the algebraic model, but apart from that, the data that was generated was very similar. And in terms of the CFD results, I think there's one key finding that I want to highlight, which kind of helps to inform the following slides, and that's the difference in the central and the peripheral lung deposition. So as you can see, for patient 001, which was the cystic fibrosis patient, we get higher central deposition compared to peripheral. And for the healthy volunteer patient 2, or subject 2, we see that that's the opposite. And actually, whether that's something general to the CF condition is something we can't tell from any 2 patients, but it's the sort of thing that we'd want to consider once we were doing a larger scale study with these things. So we use this to parameter or to input into our PBPK model. But then what we did next was to look at the central lung epithelial area under the curve. So this is a prediction of local pharmacokinetics in the lung, which of course the PBPK model allows you to do. And we ran a sensitivity analysis of this parameter based on different considerations for the cystic fibrosis disease state. So at the bottom in red, you can see the healthy volunteer. So this is the central and peripheral lung AUC, sorry, for the healthy volunteer. The blue bar is when we consider deposition in the CF patient lung geometry. So only that's changed. And you can see the expected swap between central and peripheral that I showed you earlier. Now, the green is what happens when you change the mucus thickness. So we know that in cystic fibrosis, there's a thicker layer of mucus in the lung. So we were able to just increase this as a parameter in the model to make it representative to see how that affected things. And interestingly there, it almost eliminates the difference between the healthy volunteer and the CF patient deposition differences. And it actually exaggerates the difference, sorry, in the central lung, and for the peripheral lung, it exaggerates that difference. Now, of course, this is only for 2 patients, so it'd be good to see this on kind of more of a population level. But it does start to show you how you can bring in disease state considerations to bridge data from healthy volunteers to patients within an alternative approach. And then at the very top, we also have mucociliary clearance. So we know that because of the thicker, stickier mucus in cystic fibrosis, mucociliary clearance is inhibited. So we can, in the model, we can reduce it or switch it up. And here, what we saw was that this actually has very little, makes very little difference to the model predictions of local pharmacokinetics. And we believe this is because amikacin is a fairly soluble molecule. We're using spray-dried particles. They dissolve very quickly. So actually, there's not much chance for mucociliary clearance to have an effect anyway. So, I guess following on from that and starting to wrap up now, at the beginning, I showed you 4 key attributes, which I believe are important to building an in silico methodology into an alternative bioequivalence approach, which were endpoints, product, patient, disease state. In terms of endpoints, I think it's really important to start to consider at the outset of your modeling what your product is trying to achieve so that you can figure out what endpoints to look at. For example, if you've got something that's highly soluble, you might not need to worry about doing the PBPK modeling part, and just knowing where something deposits in the lung is potentially a candidate. One thing I would note is that what we've found quite recently when we've looked at molecules like the quaternary ammonium bronchodilators from Walenga, that receptor binding is quite important. So to reproduce even systemic PK in the CFD PBPK approach, we actually need to take into consideration receptor binding, and that's something that's quite interesting and potentially gives us a route into an endpoint, because you can start to look at the receptor binding kinetics as well. In terms of the product, I think we all know that it's important to identify the right key performance attributes. And as I showed in the fluticasone propionate example, we can use sensitivity analysis within a PBPK, CFD, or algebraic deposition modeling approach to identify what those most important parameters are, both for systemic pharmacokinetics and for local pharmacokinetics as well. In terms of the patient, we have different mouth throat geometries that we can use in vitro. Sensitivity analyses in terms of breathing profiles is very important, because we know that for both inter and intra patient dosing variability, the change in breathing profiles is very important. And then, of course, there are physiological differences due to different patients, either within the healthy population or within the patients. We can inform this from CT scans, CFD and CT scans, or making adjustments to lung models in semi-empirical or algebraic approaches. And then finally, the disease state. I think the example I gave highlighted this fairly well, where we can start to introduce those levels of variability between healthy volunteers and patients into a CFD, PBPK, or algebraic deposition model, PBPK approach, to understand how a drug that's taken by a healthy volunteer may elicit a different pharmacological effect in patients, just due to those physiological differences between the two groups. So in terms of what the study would look like, based on the work that we've done so far, the first, of course, is to look at the model selection. What's the endpoint and what models produce those endpoints that you're interested in? Then preliminary characterization is important, where you can gather supporting data and model input data to parameterize and input into your models. Sensitivity analysis, I think, is probably the most important fact in all of this, because once you've brought everything together, you can start to answer the question that Rob brought up in his opening this morning about how different is different. If you know that you have a difference between a test and reference product in terms of its aerodynamic particle size distribution, you need to understand what impact that's going to have on both the local and the systemic pharmacokinetics in healthy volunteers and patients. And this holistic modeling approach, which brings all the different aspects together, can start to answer those questions. And then the final part is that in silico trial design. So in my cystic fibrosis and healthy volunteer example, there were some differences, and for 2 patients or 2 subjects, it's not necessarily clear whether they're due to the disease state as a whole or whether it's just variability between 2 human beings. So making sure that you design a correct in silico trial design, which takes into consideration not just point estimates but variance as well, is a key part of the process. And one of the advantages of in silico techniques is you can do power calculations relatively easily by running simulations over and over again. Now, for CFD, that might not necessarily be feasible, but there are lots of different methods of getting the information a lot quicker. And actually, Jan tomorrow is going to talk about some new AI-based things that are making that even easier. So yeah, takeaway messages. Important things are identifying your right endpoint, taking into consideration product variability, patient variability, and also disease state specific things. And I think sensitivity analysis, like I said, are one of the most important aspects of pulling this off. And with that, I'd like to thank my collaborators at Nanopharma Fluida. Lots of interesting discussions have gone on in the last couple of years into this topic, and I think we've come a long way. And I'd like to thank you all for listening. Cheers.